Fritz-Eibel of MolecularMD.
Good afternoon, and thank you to CMS for allowing us the opportunity to share a little bit about our companion diagnostic that's focused on treatment-free remission in CML patients. My name is Fritz-Eibel. I'm Senior Vice President at MolecularMD, and I'm joined by Marisa Pierce, who will help field questions that I can't seem to tackle myself. But we are a company that is based in Portland, Oregon, with a focus on building precision medicine assays. We'll start out with CTAs, clinical trial assays, put them under design control, and get them in a position where if they need to be submitted to the agency and in the label of a drug that they're ready to go. So this is an overview of what I'm here to talk about today, but I thought it might be interesting to talk about CML and give you a little bit of background. So the 8-year survival rate for CML back in 1983 was less than 15%. Where it stands today is 87%, so we've made some big headway. The NCI is a big reason behind that. They funded research for imatinib. And Dr. Brian Drucker in 1998 opened a clinical trial that really changed the face of the way things are done for CML patients, and it started targeted therapies. So in 2001, with the FDA approval, patients with CML, Philadelphia chromosome positive, were able to really have an impact. And those that went into remission for 2 years now have the same life expectancy as somebody who does not have CML. So huge advances, really kind of the start of targeted therapies. Pharma has continued to work towards the improvement on this tyrosine kinase inhibitor. Imatinib, also known as Gleevec, there's now a whole set of second-generation TKIs, and they have a more durable response, and they have a better impact on patient outcomes. So big advancements there. One of the things, though, that has started to come up as a novel treatment paradigm is this concept of going off drug, known as treatment-free remission. So this is the concept of being able to completely go off drug and never come back on it. And so it was back in 2012, Novartis came to MolecularMD and said, we would like to have a clinical trial assay to match up with our second generation TKI, nilotinib, also known as Tasigna. And we want to monitor really deep responses to our second generation TKI, and we need something that is going to have precision and accuracy and get to a MR 4.5. So traditionally, labs have been testing around MR 3.0. That's good enough for imatinib and monitoring CML patients. So we took this on and built the assay, and when we embarked upon this, there were a lot of challenges, but at the end of the day, we were able to get it done and really build out two very key trials in support of Novartis, the ENES Freedom and the ENES Stop trial. So the Freedom trial was treatment-naive patients. The Stop trial were patients who are on imatinib. And so we're looking to support both of those arms. The FDA considered them significant risk trials, so we submitted an IDE and all the work behind that to include building out a modular PMA in support of having that companion diagnostic. And at the end of the day, the project cost to run the trials, to build the diagnostic, it exceeded $26 million. So it wasn't a small effort, and a lot of people worked really hard at this at both companies. But it got us to a really great point in December of last year. Tasigna received a label update for treatment-free remission. It's the only second generation TKI that has that in its label. And MolecularMD received FDA authorization for the MRDx test as its companion diagnostic to determine when to initiate treatment-free remission as well as to monitor it. So it is in the package insert for Tasigna, and it states that a patient has to be on the drug for 3 years and have achieved a sustained molecular response of MR 4.5. The test is the only authorized test to be used for the initiation and monitoring of patients attempting TFR and can be found on the FDA website at fda.gov/companion diagnostics. So, MR 4.5 is really important, as well as having reproducible sensitivity and specificity, because you don't want the patient's BCR-ABL level to bounce around, because then it makes it hard to make a clinical decision on whether someone is relapsing or if they're still in line. So we've had to do some things that go above and beyond the traditional BCR-ABL test that there is a CPT code for, and that's why we put in for this PLA code. We require 5 mils of peripheral blood and EDTA. That creates a little bit of a logistical issue, because that's usually more than what most laboratories collect. It also has to be drawn and shipped at 2 to 8 degrees, and it has to keep that temperature. And from the time of draw to the time of extraction, it has to be done within 48 hours. So the logistics are not trivial in doing all that. But then also, in order to get the accuracy and precision, the test is built a little bit differently than what other tests have. Let's see if I can forward here. The test itself runs the patient samples in duplicate. It also has 15 calibrators per plate, and it has 10 controls per plate, to include one control that is at MR 4.5. So if that control fails, the plate fails. So we ensure that the patient samples are measuring at a depth of MR 4.5 consistently. The market realities and the implications for TFR are worth mentioning here. Currently, the CAP proficiency testing is done at just MR 3.0, and it does not go as deep as deep molecular response, which is MR 4.0, or where we would want it to go, 4.5. So we need to advocate for change with that. Also, third-party market research indicates that 33% of the U.S. labs test to MR 3.0, and 46% test to a level of MR 4.0. So while that's sufficient for second-generation TKIs and just monitoring of patients, it's not sufficient for TFR and what has been borne out in the NS Freedom and the NS Stop trials. So as mentioned earlier, the MRDx test requires significant changes to sample logistics as well as workflow, and it has an impact on overall throughput. And a worthwhile example is one of the large reference labs that commands a large market share of BCR-ABL testing is able to do 80 samples in 196 well plate. That is compared to what we have, which is 17 samples per 96 well plate. So what that means is potentially, you extrapolate, that's about 5 times the labor. So it's not a very high throughput test, but it is required in order to get the accuracy and precision and reproducibility. So the MRDx test has specifically been developed and indicated for use with TFR initiation and monitoring, and it's not necessarily meant for broad CML patient monitoring. You see here that I've put cost per sample and total cost at $90 a test. This represents the cost for us to manufacture the test and to procure the reagents for the Promega Maxwell CSC, which is what's used for nucleic acid extraction. If we look at our cost accounting and what it costs for our all-in the CLIA lab that we have in Portland to run the MRDx test, the result is about $281. It's worth noting that this does not include capital equipment or a plate failure rate that is around 5%. So with that, what we're looking to do is have a multiplier on the 81206 CPT code of 2.5, requesting $506.05 for the MRDx test. The reason for that is it is an FDA authorized companion diagnostic. A significant amount of work went behind getting that done. It's also super important for patient safety and making sure that TFR is something that continues forward and we work towards a cure for CML. There was a lot of investment in this, but nothing good is ever easy. And finally, the test protocol requires, as I mentioned, some challenges with logistics and also the workflow within the laboratory. So, thank you.
CMS, any questions? No. Panel, any questions from panel folks?
This is Brian Lloyd, a panel member. Thank you for your presentation. Tell me, I heard you mention that the test and the depth of monitoring and the frequency of monitoring located in the package insert for Tasigna. Is it also included in the package inserts for the other TKIs for CML?
It is not. Right now, Tasigna is the only TKI that has an indication for treatment-free remission. The NCCN guidelines highlight with second-generation TKIs the need to do monitoring and all, but Novartis was, I think, the first mover in trying to do treatment-free remission with their TKI.
And does the NCCN recommend or suggest or consider using this type of testing for the non-Tasigna type of therapies, TKIs?
I'm going to refer to Marisa on that. She's closer to the NCCN guidelines than I am.
The NCCN guidelines suggest using a test that can reach MR 4.5.
Thank you.
Hi, this is Michelle. I have an additional question. There is another FDA cleared test that I believe is cleared to MR 4.7, although it's not, I don't believe it's specific for any particular drug. What would you say would be the additional benefit to this test that justifies the additional cost over a traditional quantitative BCR-ABL assay that could reach the same endpoint?
Okay, good question. Yes, there is one other FDA cleared test that is a Asuragen's test. It does not have the indication for treatment-free remission. It is not in their label. I think probably the big distinction here is having a control at MR 4.5. To claim that you can go deeper than 4.5, our assay actually does that too. But our cutoff was set with Novartis at 4.5, and we can demonstrate that in a consistent manner with the use of our control. Others cannot.
Okay, anyone else?
This is Brian Lloyd. Can I ask one more question?
Sure.
Is there any difference at the initial testing for establishing this treatment-free period versus the use of the tests in the monitoring and following aspect of the utility of this test?
You know, that's a good question. We've been talking with Genoptics about that. I think one of the challenges that we're working on is how do you bridge from one BCR-ABL test to the MRDx test? And we're working to try to solve for that. And part of that is looking at the WHO standards, the controls we have, and trying to make it so that there is a way to reconcile the two. I do think this poses a challenge and one that we'll work towards, but the way in which things are set up within the package insert for Tasigna, and it's probably best that Novartis speaks to that rather than me, but the desire would be to use the MRDx test as a patient is attempting treatment-free remission.
Thank you for that. But if a doctor were to use the MRDx test as establishing eligibility for discontinuation of Tasigna, the exact same test is used to monitor, to maintain that they can stay on that discontinuation status. Is that correct? There's no difference in the test in those two different scenarios.
Yes, sir. Same test.
Okay.
Same test, and there's a different testing regimen that occurs each year. There's a need to test more once someone comes off drug, and that's logical. That's for safety and efficacy. But then it goes back to a similar regimen that is being done with the traditional BCR-ABL test, which is 4 tests a year, so every quarter.
Thank you.
Thank you.
So this is Michelle again. Maybe I can call one of my panel members to phone a friend, Aaron from AMP. I know that there's been a number of BCR-ABL tests that are LDTs in the past. Do you have a sense for how many of those are scaled to the international scale, and what the range of cost would be? I know AMP had done a cost analysis of a number of molecular pathology procedures in the past, and that's why I'm asking.
I don't know that that's anything that I'm able to fully share. We've done some market research on it, but I don't consider it to be robust and something that would hold up with this audience. With laboratory developed tests, you're not always able to see what's there unless you go in and look at the lab report, and then you poll everything and see who's reporting on the IS. The international standard is something that's become more commonplace. So you see that across many of the LDTs. 5 years ago, not so much. Bruce?
Bruce Quinn, very quickly, it sounds like this was described as a sole source test in Portland and also as a PMA approved test, so it would qualify as an ADLT, which would fast track your pricing process.
Point of clarification. The FDA at the last minute decided to make it a 510K. So it isn't a PMA. It is an FDA authorized 510K test, a companion diagnostic. And we do run it in Portland, but we have an arrangement with Genoptics that they're going to be the exclusive laboratory for us. And we did offer the test in Portland simply because we needed to make sure we were taking care of patients. Our core business is not in CLIA testing, but we have the accreditation, and so we used our lab as a bridge until we could get Genoptics up and running and able to do the test. So thanks.
Okay, our next speaker, we're going to switch it up a little.